All right, thanks for coming by. My name is Garrett, and I'm an in-house scientist at ACEA Biosciences. And, today I want to introduce you to the NovoCyte, the new flow cytometer for everyone. So just a quick background about ACEA Biosciences and who we are. We started in 2004, and it was basically built on the technology of a label-free real-time cell analysis platform. And we have many different models that we've launched since 2008. We were fortunate in basically a year ago, we launched the NovoCyte, which complements our cell analysis platform, where we're able to label cells and analyze them this way. So that's what I want to introduce you to today. The NovoCyte flow cytometer, it's pretty small. It's a benchtop cytometer, so it's not going to take up a lot of space. And what's really nice is we can measure up to 15 different parameters, including forward scatter and side scatter. So that's 13 fluorescent channels. We have a very large dynamic range, which makes for planning and running your experiments. It makes it a lot easier. And we have a lot of automated features that I want to touch on today. So some of the main benefits of using the NovoCyte includes it has high performance and sensitivity. It's very small, so it's not going to take up a lot of space in your lab. It's flexible. It can be upgradable anywhere from 1 to 3 lasers and anywhere from 13 to 13 fluorescent detection channels. The software is very intuitive. There's less training time involved, and this really makes running your experiments a lot easier. And lastly, we offer the best price, where you can do your successful research at an affordable cost. So real quickly, this is an overview of our the optical layout, where we have our lasers lined up here. These are lasers made by Coherent, and they're expected to have a very long lifetime. They're in a fixed alignment, so you don't need to constantly align your lasers for the optimal signal. And these are also upgradable. So if you only choose to have a machine with 1 laser right now, you can definitely upgrade this to 2 or 3 lasers over time. And that's simply just a service call. You don't have to send the machine in. So we have our lasers here. They come and interrogate the cells in the flow cell, and then we are able to detect our signal here with our PMTs. So this is customizable. We can upgrade the number of PMTs as your research sees fit. One thing that's really nice is that we have an optimized PMT voltage. So these are very high. These PMTs have a high sensitivity, so they have 7 decade dynamic range. So we've optimized the PMT to where you don't have to adjust your PMT voltage gain up or down to get the peak in the right spot. We have enough range of signal detection that we're going to be able to see changes no matter what. This kind of shows you an example here. So in this plot up top, this is a competitor's instrument with a 4 or 5 decade dynamic range, and you can see that the dim signals and the bright signals are smashed up against the axes. Normally, if you had a sample like this, you would move the voltage gain down or up to get the optimal position of your peak. However, with the NovoCyte, since you have 7 decades, you're able to detect the dim signals and the bright signals in the same plot, and so it just makes your analysis a lot easier. So we've optimized these PMT voltages so you guys don't have to do any additional adjustments. We can acquire rates at up to 35,000 events per second. And because of this, because you're not adjusting the PMT voltages, it really makes compensation a lot easier, because you don't have to, because you can compensate your samples after you run your experiment, and you don't have to adjust these voltages for every one of your parameters. So this is an example of our resolution and dynamic range that we offer with the NovoCyte. On the left-hand side is NovoCyte running whole blood sample, and on the right-hand side is a competitor's instrument. And what I want to point out in this top chart is we're able to distinguish lymphocytes and eosinophils in the same plot, whereas we cannot do that with the competitor's instrument, because we're looking at the lymphocytes here at the expense of the eosinophils. If we want to look at the eosinophils, we have to move the voltage down at the expense of viewing the lymphocytes. And then also I want to show you that we're able to detect lymphocytes, monocytes, neutrophils, and eosinophils based on the forward scatter, side scatter resolution with the NovoCyte, whereas with a competitor's instrument, we see lymphocytes here, but we have monocytes and neutrophils all in the same plot, so we're not able to resolve these based on forward scatter, side scatter. They would need additional markers. This is some resolution and sensitivity stuff that I'm actually not going to go into. I'm going to let Ryan take care of this. I do want to point out that in the when looking at size resolution, we can detect anywhere down to 0.21 microns based on the particle size standard beads. Another very, very useful feature with the NovoCyte is we have a syringe pump driving the fluidics, and so we're able to obtain direct cell counts from all of our samples. And so if you're interested in the amounts of lymphocytes in your sample or the amounts of white blood cells in your sample, you're able to get this with every single sample that you acquire with the NovoCyte. This is just a graph showing that we took certified bead concentration particles on the x-axis and used the volumetric absolute count obtained by the NovoCyte on the y-axis, and they align pretty nicely. Their R-squared is 0.999. So this makes also, if you want to look at proliferation in your samples, you're able to accurately count these. And lastly, the NovoCyte offers a lot of automated system functions, so you don't have to spend a lot of your time doing routine maintenance, routine cleaning, or monitoring your fluid levels. Startup and shutdown is a push of a button, so you're not, when you're shutting down the system, you don't have to switch between bleach and water and do that for a certain amount of time. You can push the button and you can walk away. Cleaning of the SIP, you can set the machine to do that between every sample or at the end of your experiment, whichever you prefer. We have a lot of maintenance features that are a simple click of a button, such as debubble or an unclog feature, back flush, priming the system, all that's just a push of a button. Our small fluidics cart that sits next to the machine can detect if your sheath fluid is running low, if your waste fluid is getting high. And then lastly, we have automatic QC functions. So you can run the QC beads every day, and you can monitor these changes over time. That's what I'm showing here. So there you have a Levy-Jennings plots that are automatically done for you, and you can set the time period that you want to analyze, and you can look at the system performance over time, so you can see if your system is behaving just like on the day it was installed. This is a couple screenshots of the software, NovoExpress, that comes with the machine. I already told you about automatic compensation. We can do quick compensation, which is just a manual scroll bar that we can adjust to compensate our samples on the fly. We include a cell cycle analysis module, so you don't have to make these precise gates on your G1 and your G2M peaks. And lastly, we can do these automatic reports that are generated, and these are completely customizable. You can put 3 plots here on the top, or you can put 33 plots if you want. So it's just a really nice and convenient feature. Here's some of the toolbars that you'll find in the software. You can see, we can import or export FCS files. You can do absolute counts, just a push of a button, and it will add a statistic to your table. I talked to you about the QC buttons, the fluidics maintenance buttons, and then our experiment manager. So when you want to apply gates from one sample to your entire group, it's a simple drag and a drop. It's a very Flojo-like analysis. This is just a quick example of a 13-color immunophenotyping profile that we've run before. I'm not going to go through all of these plots. I will be presenting 2 posters tomorrow if you want to look at some other different types of cell analysis, such as eosinophil analysis, other white blood cells, as well as stem cells. Oh, and lastly, I want to talk to you. So we include a NovoSampler that's you're able to run 96 well plates, 24 well plates, actually a 24 tube rack, which I find very convenient with the fax tubes. And you can do this for high throughput analysis. So you can set up your experiment to just run from sample to sample, and it will automatically run these samples without you being present. There's built-in automatic self-alignment procedures with this, and it's very interchangeable with a single tube loader, so it's very easy to do. So thank you for this. Let me give you this introduction. Please stop by our booth if you want more information. We're at 1007, and I have 2 posters that I'm going to present tomorrow afternoon. So I'll hand this off to Ryan, and we'll be happy to take questions after this.
All right, thank you. Thanks for coming. And like I said, my name is Ryan Duggan. I'm from the University of Chicago. I run a core facility there. We have many different instruments, a dozen or so that are currently working in our lab. So we have also the ability to test lots of the new instruments that come out on the market. And what I'm going to explain for you today, or go through today, is kind of our battery of tests that we use when we're testing out new instruments. It's a standard set of tests that we run across every instrument, and that way we get some good performance metrics on each of them, so we can kind of see whether they'll be useful for the applications that are being run in the core. I just want to begin by framing the discussion here that might be familiar to you in a few different ways. One is, if you're in the market for purchasing a cytometer, this type of information will be useful for you in trying to make those decisions. The second would be if you have access to a core facility, and let's say that core facility has a few different types of instruments, you might want to do some of these performance type tests to see which one would be best for your applications. Also, if you're involved in a multi-site study, and you have collaborators at other institutions with different instruments, perhaps this would be some kind of testing that you'd want to do to make sure there's kind of a baseline performance of all the instruments involved. And then lastly, if you just want to get a better idea of the performance and quality control of your instrumentation, we can help show you some of the metrics that we use. So the question then is, what can we do? How can we evaluate the performance of a flow cytometer, really using some quantitative metrics so we can put some numbers on this information? So what really matters? And we're going to get beyond kind of the basics, and we're going to make the assumption that things like types of lasers and the number of detectors, the event rate, those all are sufficient for your needs. Let's move beyond that and say, how can we differentiate the various instruments that we're looking at? And the one thing that we'll do is we'll obviously look at performance of the instrument, but really the key part here is that we want some metrics, some quantitative assessment of these performance. So at the University of Chicago, we've developed kind of our core battery of key performance indicators that we use on every instrument. The first being fluorescence sensitivity and resolution. Those we use things like multi-intensity beads, as well as a metric that we developed called the Q norm. And this is really going to impact the ability to resolve dimly stained populations. The next is looking at linearity. For that, we use kind of the standard chicken erythrocyte nuclei protocol. And issues with linearity can affect things like compensation or fluorescence quantitation, or if you're doing cell cycle analysis. The next thing we focus on is carryover between samples. And obviously, if you're trying to detect a rare population, this could have significant impacts on your ability to do so. And then lastly, there's a set of intangibles that we like to look at. Ease of use, the software, the hardware, interacting with the instrumentation. Obviously, this could affect the efficiency of your workflow, but also if you're in a core facility, this could affect adoption by the users. Some users might just say it's too difficult, let me move back to the platform that I'm more comfortable with. So the current norm for instrument performance from a fluorescence intensity standpoint is really an idea called the detection threshold, which has been around for many years. And this essentially looks at a bead set that has fluorophore immobilized on the cell surface at varying amounts. So you're able to convert the relative fluorescence scale that we normally get on a flow cytometer into a scale that now is molecules equivalent to the soluble fluorophore. And these beads are NIST traceable, so the National Institute of Standards Technologies kind of keeps tracks and governs these. But really, they're only available in 2 colors. And what happens here is you can extrapolate down, and the detection threshold, in essence, ends up being the y-intercept of this line. So you'll get values like our instrument can detect fewer than 100 FTSE molecules. And there's some issues with that I'll explain in a minute. The other common thing is to look at 8-peak fluorescence beads. And these are a bead set that's commonly purchased from Spherotech. and this gives you 8 different fluorescence intensities across many of the different channels. The current norm really gives you, at the end of the day, somewhat of a meaningless metric in sensitivity. Saying that you can detect fewer than 100 molecules of fluorescein on your instrument is kind of a moot point. Autofluorescence tends to be much greater, maybe an order of magnitude brighter than the detection threshold. So really, we don't care so much about that absolute threshold in terms of sensitivity. What we really care about is fluorescence resolution. Being able to detect something that maybe has 550 units of fluorescein from something that has 500 units of fluorescein. That's really the important part. The other thing is that these beads are not really available outside of a very limited set, like FITC and PE. So because an instrument has good FITC and PE sensitivity, does that necessarily mean that it's good for Alexa 700 or some of the brilliant violet dyes? No, we can't really make that jump. The 8-peak beads have the deficiency of just not really being quantitative. So you can display the picture of the 8 peaks, but really, what is that telling you? What real life information is that telling you when you're trying to build a panel that you're going to have good sensitivity? But nonetheless, the 8 peaks are important. They can kind of give you some gross issues, can display any kind of gross issues that you might have in detecting fluorescence across the board. And so this is the data from the NovoCyte. Again, qualitatively, you can see that, yes, we can resolve 8 peaks across many of the channels you'd expect to. Typically, in the far red channels, the resolution is not that great on any of the instruments, and that's more of a dye chemistry issue than an instrument detection issue. But this is pretty much what you'd expect across the board from 8-peak beads. And to prove this point a little bit further, the utility of 8-peak beads, here's just some data we have from 2 different instruments. And this is looking at the channel that we typically look at PE-Cy7 in. And so what we have here are the 8-peak data, and usually we look at this low end, we say, okay, great, you can't resolve 8 peaks. You can resolve, 1, 2, 3, 4 peaks on each instrument. So the question is, which instrument is better? And I think everybody here would be hard pressed to pinpoint what instrument is better in this case. They look pretty much the same. If we just do one additional test, and this is a bead set that is stained with a PE-Cy7 antibody, we can look at the resolution of the 4 peaks like we should have on this instrument versus 1 peak and then kind of everything smashed together on the other instrument. The same exact instruments, same laser excitation, same bandpass filter essentially. The only difference is we're using a different way to try and differentiate the 2 instruments. And so in this case, the take-home message is that embedded dyes in a bead, like you have in the 8-peak beads, do not necessarily give you performance indicators of actual fluorescence, organic dye fluorescence that is mobilized on the surface of a bead, or in this case, a cell. And the 2 cannot be directly correlated. Now, there are other ways that people have looked at fluorescence sensitivity. I think another very common one is the Q and B method, Q being the quantum efficiency of detection on a given channel, B being the background. And so you can see the differences in Q here. Very good Qs give you good resolution of peaks. Poor Qs give you poor resolution of peaks. But in the end, these numbers don't really mean a whole lot. Sure, they're quantitative, but if I were to tell you I have a Q of 0.03, is that good or is that bad? It's hard to know. So what we wanted to do is kind of bring all this information together and come up with a metric that is understandable and readily applicable to your science. And what we came up with is this value that we've dubbed the Q norm. Q norm stands for quantitative, results in a meaningful number. It's normalized because we can run it on any given channel, on any instrument, normalize the data across them so that we can quantitatively look at differences between those instruments. It's based on the overlap ratio of unstained lymphocytes and a dimly fluorescent bead. And metric because I needed another letter to make it a nice sounding acronym, so I put metric in there to give us Q norm. And at the heart of this assay are a set of beads here. These are beads that are labeled with varying amounts of antigen binding points on the bead surface. We use a product from Bang's Labs, the antibody binding capacity beads. They're called quantum simply cellular beads. And these beads are stained with whatever antibody you want. So now I can choose whatever fluorophore I want. If I want to specifically test Pacific Blue, I test that. I'm not limited to the FITC and PE MESF beads. And then those are run on the instrument, and some calculations are done to give us that Q-norm value. So this is essentially the basic protocol of the assay. It's very simple. It's just beads plus antibody, run them on the instrument. We run, along with the beads, unstained lymphocytes as kind of our background, standard background for each of the assays that we run. And the math is a little bit convoluted, but I'll just explain it this way. So here are our 4 peaks from the quantitative ABC bead set. This allows us to take the relative fluorescence intensity scale and convert it into a new scale that shows us the number of antibodies bound. So now this is a quantitative scale that gives us number of antibodies bound. If we can imagine this peak becoming dimmer and dimmer, we stop at the point where the 10th percentile of a dim bead is equal to the 90th percentile of our unstained lymphocytes. So that's the overlap that we're looking at. So that's what we're going to say at that level we'll still be able to resolve. Any less, we can't resolve anymore. A bead that has a 10th percentile fluorescence intensity value equal to the 90th percentile of unstained becomes, in fact, the Q-norm. And this value that we've now generated is the minimum number of antibodies that needs to be bound to a dim positive population that can be resolved from background or autofluorescence. Okay, so now this is a real world number that we can work with and compare across instruments and see what is, in fact, that resolution threshold that we're working with. So here's some data, and this is essentially 5 channels that I try to test on every instrument. Of course, I could do more channels since any antibody that I can conjugate to a different fluorophore would work. This represents all of the instruments I've tested in the FITC channel. You can see there's quite a big spread, some of them better than others. The gray box represents kind of that middle 50% of that group. And the blue dots shows you how the NovoCyte performs in those channels. So lower numbers is better, because that means our resolution threshold is lower, so that's good. And so you can see in like the FITC channel, for example, the NovoCyte is performing well. And this is not just instruments with similar capabilities and similar price level. This is everything. I mean, these are industry standard, top of the line performers that are mixed in this group as well. All of them that we've had the ability to test at University of Chicago. The other nice thing that you get from this data, which you guys are probably already familiar with, is that you can see pretty clearly that PE and maybe PE size 7 and APC are really going to be our good performers on the instrument. Those are the channels that we're going to use for the populations of cells that we're trying to resolve that might not have a lot of antigen present on the cell surface. Conversely, things like FITC and maybe Pacific Blue are not that great. So you're not going to put your low abundant antigen presenting cells in those channels. Now, of course, if I change this to, say, Brilliant Violet 421, we might have a whole different story. But that's the beauty of this assay, is I can use whatever fluorophore I want and test them specifically. So to put this into perspective, CD4 is expressed on human lymphocytes. It has between 45,000 and 50,000 binding sites on your average CD4 cell. So you can see on most instruments we're able to at least detect 10-fold less than that. Some instruments we can detect even less than that. And CD45 is even brighter on the lymphocytes, is about 180,000 binding sites. This is showing you the difference between CD4 staining on a channel with a really good Q-norm and a channel with not so good of a Q-norm. So this is the same CD4 antibody staining with Pacific Blue, which is the blue histogram, and PE size 7, which is the red histogram. So obviously on the PE size 7 channel, our separation is so good that we'd probably be able to pick up many fewer antibodies bound in that channel than we would on the Pacific Blue. Pacific Blue has this characteristic, a little bit higher background fluorescence from that violet excitation, and maybe not as good collection of the PAC blue fluorescence, the specific fluorescence. Okay, so that's fluorescence sensitivity and resolution. So that's one of the key areas that we look at. The next ones are a lot less involved, so I'll go through them quickly here. But linearity is also an important thing to check on the instrumentation. We use chicken erythrocyte nuclei stained with a DNA binding dye, typically PI, to test this out. And deficiencies in linearity can really impact things like compensation, or if you're going to do fluorescence quantitation, you'd obviously have an issue there, as well as if you're doing things like cell cycle. The data looks a little bit like this. So CENs have this characteristic that they like to form these aggregates. So you get this characteristic peaks at 1 nuclei, 2 nuclei, 3 nuclei, and so on. And since the DNA dye binds stoichiometrically, we get a very well-known and calculatable value for each of those peaks. So because of this, we can actually draw a theoretical line, which is this red line here, and then look at where the instrument is putting those peaks, using the median of each of those peaks and putting that into the graph as well. And you can see linearity across the scale. This is going up to 12 nuclei aggregates. Deviates from the theoretical by not more than 3%, even all the way up at the top. So we have good linearity across the board, which means that would translate into pretty reproducible compensation, so we don't have to deal with additional issues with linearity affecting compensation. Another test that we do is focusing on carryover between samples. And for this test, we use the NovoCyte's autosampler on the instrument and sampled directly from wells of a 96 well plate. And I used the standard washing protocol that was set up by default, so without any optimization or anything like that. And I used the same CENs because I have them already from the previous test. And I run, in the first case, I run unlabeled, unstained CENs. So you can see we get this nice blue peak, but there still is some junk that comes up in the higher scale. These histograms, by the way, I should mention, are normalized to the mode, so that's why even the rare events here are showing tall peaks, just so we could see them a little bit more easily. So you can see here we get about 100 counts in our positive in our background. And then when I stain the CENs with PI, you can see we get a nice shift. These are bright, and now we have 200,000 events out there, and these guys are just debris or unlabeled events in the bottom. It does a wash after that, and then I put back the unlabeled CENs and run them again. And so you can see here, whereas we had about 100 background events in the control group, now after the wash we have about 370 or so, maybe let's call it 400 events. And these are actual events from the prior sample that have transferred over into the next well. I'll just mention that I like this test because every instrument I test fails. And so it just really reiterates the need to optimize these types of things when you're on the instrument. So after the first wash, then we do another wash and run them again. So I have another well of unstained CENs, and now it goes back down closer to baseline. So after that second wash, we've gotten rid of all that carryover. But there's one additional carryover that we have to worry about. Not necessarily the cellular carryover, but the carryover of the PI, the dye itself. PI is notoriously sticky and difficult to get out of lines, so that's always a good test for me as well. And so for that, I focus on the intensity of the blanks, of the negatives. So you can see the blank is initially around 2,000 on the scale here. And then I run stained stuff, and then I run unstained stuff again. And so you can see now the unstained population has jumped up to 4,000. And this is actually PI that has transferred from the first well, from the second well, sorry, the PI stain, to the third well after a wash, and has actually stained the unstained cells just a little bit, just to bring them up a little bit. and that's actually dye transfer from well 2 to well 3. Again, one additional wash brings that back down to baseline, and that's usually what we expect. Usually the 2 washes to kind of get rid of this is kind of the standard. What this means really is that if you were doing kind of a rare population type assay, that you'd really want to optimize the washing procedure between your wells. And thankfully, with the NovoCyte sampler in their software, you can actually program in additional washes between wells, so it's not really an issue. The last thing that we typically look at are what we just call intangibles. So these are kind of things that you look for over many years and many instruments that you've tested. This is really focused on usability, both from a software and a hardware standpoint. Again, this will really impact the efficiency of your workflow. If you got a really bad usability, it'll just take you longer to do things, and you won't actually enjoy running stuff on the instrument anymore. So that can be a pain as well. And there's a few things that I'd point out in terms of usability on the NovoCyte system. The first is that really usability is the NovoCyte's #1 feature. It's pretty clear to me, having spent the past few months running the instrument, that it's really simple to set up and run. I like to check on a metric that we use called a cold start to dots on the plots. And as the name implies, this is going from an instrument that's completely shut off, starting it up, doing any kind of startup routines, doing whatever kind of QC is built into the system, and then running and setting up your sample until you get dots on the plots. On some instruments, this can be quite long. If they have mandatory built-in startup protocols and QC protocols, sometimes it could take 20, 25 minutes to go through that whole process before you've even ran a single sample. On the NovoCyte, because they have a hardware button on the instrument itself that not only turns the instrument on, but also automatically sends it into a startup routine. By the time you're actually logged into the computer and open up the software, startup is on its way to being done at that point. Then it's a pretty simple and quick QC process. And then after that, the real key here is that because I don't have to adjust voltages, I don't really need to open up plots, a bunch of plots, to look at my populations and position them on the scale. So I could bypass the whole point of creating plots and regions and gates and just literally map out my plate, put it on, and start collecting. Then take a look at everything during analysis. I've been doing that, using that method over the past month or so, and it's worked out really well. I can kind of just turn on the instrument, run QC, throw my plate on, hit go, walk away, come back in a half an hour or whatever, pick up my data, and just start analyzing. So the workflow is extremely streamlined. And it's because of, again, one of the key features is that fixed voltages, which for the most part is going to work. 90% of the time, you throw on most of the samples that you commonly run in the lab already, the voltages are going to be fine. There could be those cases where you have a really bright cell line, or even I found if I had freshly labeled CFSE lymphocytes that are super bright, they use way too much dye, you might be hitting that upper end of the axis. There is a way to go in through some administrative settings and adjust the voltages some degree. So it's not like you're stuck at that point. You have a way out. But for the normal user, routine day-to-day stuff, you're just going to walk up and run samples and be done with it. The other thing that I'll mention here is really the auto sampler. The auto sampler is really nice. We've used samplers of all different types, from the BD HTS system to the IntelliCyte sampler. They all work fine for the most part, but usually they take a little bit more to set up, or you worry about vortexing, or you worry about sample settling, or you worry about clogging, and you can run into some issues. We have most of them. We have issues on most of them. The NovoCyte sampler is smooth. It's pretty quick, but I think its key feature is allowing you to walk away. It's allowing you to set up, put that plate on there, hit go, and walk away and have confidence that things are going to be run and maintained properly. And then lastly, there's a few nice software interfaces that are really familiar to you. So they use the ribbon style that is pretty popular with Microsoft Office users, and so that's a familiar feel. Also, some of these drag and drop features, if you're a user of FlowJo, you'll feel right at home copying samples and copying settings and plots and regions from one sample to another, or that kind of group hierarchy that's familiar in FlowJo. All those features make it a pretty easy setup. So to summarize, I would just say, really, again, the key feature of the NovoCyte is its ease of use, usability. So it's definitely easy to use, it's easy to set up. Its performance is on par with the industry leaders. I mean, it's probably not going to blow you away in its performance in terms of fluorescence sensitivity and resolution, but for that, 90% of the time application, it'll do the job for you. And obviously affordability factors into that as well. And I like to use this kind of Venn diagram for really all of my decisions, purchasing decisions. And like most things in life, there's always a trade-off, right? You can't really have that perfect instrument that sits in the middle here. And so we're trading off between performance, usability, and affordability. And there could be some nice high-end instruments that perform very well, are usable for the most part, but they're not very affordable. And so if you have lots of money, good for you, go buy them. But many people, affordability really plays a key factor. And so if you might have something that performs well and is pretty affordable, but is a little bit difficult to use. And it's probably functional at best. And I think kind of where the NovoCyte falls is in this category here. Usability, again, is key. Affordability is obviously going to be there. And it's kind of on this continuum, just knocking at the door of this middle piece here, where it's going to be able to hit that high performance that we're used to. So trade-offs are always going to be part of the decision-making process. For an individual lab, maybe user-friendliness is a key feature for you. Maybe if you're in a high-end core facility and you have lots of expert technical staff, you might be hanging out in this high-end area where you can probably get some funding through shared instrumentation grants and whatnot. Whoops, I had one last slide here. I'll just zoom through it. So that's it. So I just wanted to say thank you, and thanks to the folks at ACEA for letting me have the NovoCyte for so many months to test and play around with. It's been a pleasure. Also, the rest of my group at the University of Chicago for helping me run most of these tests. And I'll also just mention that I'll have a full review of the NovoCyte up on our facilities blog, which is at ucflow.blogspot.com. You can check that out when it's available. And thank you, and if you have any questions, we'll be sure to answer those for you. Thank you.
Thank you for the talk. I have a question about the sensitivity separation. So you talked about the human method. I think that one utilizes lymphocyte offset level as a kind of benchmark. So, but actually, offset level can be different from the genotype. Also, separation like if there is a fixed node, may change the offset level. So, how can that method can provide stable or reliable results?
Yeah, if I understand you correctly, you're talking about differences in baseline autofluorescence for different cell types or different states, like activated or not activated.
And, I think also I understand that the kind of different may provide different results.
Sure, exactly, and that's that could actually be a benefit. If you are only working with, say, macrophages, maybe that becomes your baseline from which you can calculate limits on that high autofluorescent population. So this is the reason that this can work is because the background is standardized to a specific phenotype a specific cell type, and not, it's not like I'm comparing activated lymphocytes on one day versus naive lymphocytes on the next day. That is all carefully controlled for from time to time.
So, do you recommend users to run your genome ACEA test for tomatoes?
Sure, that's definitely a possibility. That would give you, so it's really, the comparison is what matters. So if you had two instruments to choose from, perhaps this would give you a quantitative way of measuring the differences between the instrument and how that could impact your applications when you run them.
My second question is about carryover. So you mentioned that's one voltage development for your PPI carryover test, but the second also random walking slide maybe also. But I think it takes longer. So it's around like let's keep the way.
I don't know offhand. Do you know offhand how much the watches take?
It would take more than an hour to run a night.
Yeah. Yeah. Okay. Thank you.